So trauma has emerged to be now the leading cause of death in the U.S. in people under 70. As we've managed cancer better, as we have managed cardiovascular disease better, trauma is becoming ever more important. And it is the #1 cause of productive years lost. So it's a big, big problem. At our institution, which has grown to be the largest academic health system in the U.S., we admit some 12,000 trauma patients a year at our 5 level 1 and 2 trauma centers. And it's the most common reason for admitting a patient to a single service line. You don't hear about this very much in the mainstream medical journals because it's just not very sexy, I guess. So what do we know about the patterns of death and morbidity in trauma patients? Well, if you were to go back 30 years, represented here by the yellow line in these very important early papers by Don Fry, Eugen Feist, and others, they identified a triphasic death pattern, just talking about death as an endpoint, where many patients die at the scene. A few will die very early on from head injury, blood loss, and early MOF. And then there was this delayed peak where patients would linger in the ICU for about a week, become susceptible to infections, and then develop sepsis and die. Because of the development of specialized trauma care delivery units, that third peak has pretty much gone away. And what we see is a different form of morbidity. Again, Yoram touched on this, but organ dysfunction, susceptibility to in-hospital infections, and prolonged ICU stays, this persistent critical illness, if you will. And the cutoff point seems to be about 14 days. Once somebody stays in an ICU for 14 days, you know, they're going to be sick for a while. And for every patient that experiences this complicated or dies, there's another 8 patients that develop this complicated course. And this is important not only because it consumes resources and results in a long hospital stay, it's now shown that these patients go on to have additional morbidity at 3, 6, 12 months. So this is a surrogate for what the patient's going to look like 3 to 6 months later when they're sitting in a skilled nursing facility with cognitive dysfunction and continue to be and really don't have a quality of life. So this is what we're trying to impact. So what are the causes of morbidity and mortality after a patient arrives to the trauma center? It's basically 3 large categories. About half are due to head and brain injury, bleeding, coagulopathy, and its complications, and then this immune dysfunction, which actually interrelates very closely to both of these insults. And this immune dysfunction or dysregulation, we can debate what kind of term we should use here. We've moved more to dysregulation than dysfunction, accounts for a lot of the morbidity and mortality. And of course, huge efforts have taken place to try to understand this better. And I would call this the effort of the last decade, which was the effort of the GLUE Grant Consortium, which this was among the most important contributions. A paper published now, well, three years ago. It was December of 2011, looking at the conclusion of the data on our extensive gene array analysis of circulating leukocytes in severely injured patients. And I don't have the time to go through the extensive amount of work that was done here. But one of the things that this particular paper did was refine the model of what has been used to depict the inflammatory response following injury. This model established by Fred and Jean Moore in the 90s, which proposed that there was a pro-inflammatory response followed by a counter-inflammatory response, and many of us used this figure for years, and it was depicted again. Again, these are the figures that you would see in the review papers and the textbooks. And this interesting model proposed down here largely by John Manick, Jim, Eugen Feist, and others, but that began to propose that actually there was a more of a simultaneous dysregulation, a pro-inflammatory side, and a change in the adaptive immune response here, which I think was clairvoyant in a way because it did better represent what was going on. But many of these models have suggested that you have to have a second hit. And I'm not sure that's true. I think the initial insult probably is enough to get the thing going. But what did the GLUE Grant paper tell us? At least looking at the transcriptome as measured by the best AFI arrays that were available at that time, so it's biased based on the transcripts that could be identified on that gene chip, this is what appeared to be happening. Patients that would go on to all the patients developed a increase in the expression of transcripts that would segregate into pathways associated with the innate immune system, and a simultaneous suppression in transcripts that segregated more towards the adaptive immune system. Patients that had an uncomplicated course, that is, they got better and didn't spend a long time in the ICU, resolved within about 7 days. Those that developed a complicated course had an exaggerated early change and then a persistent change as well. But they didn't talk, those that did the analysis, didn't really talk very much about the types, specific gene pathways involved, just that these changes took place. So it, again, refine the picture somewhat. But there were important limitations. It was, I think, a great study. It was conceived around 2000, 2001, and based on the technology and the views within the biomedical field, it really did use the most advanced technology at the time. But it only studied the severest of the severe. I mean, these were patients that were predicted to have about a 30 to 40% mortality. Sampled a single compartment using really a single assay, that is the gene array, and sampled circulating leukocytes. There's a lot of immunology that takes place in other sites. And again, the primary focus was on mRNAs, and it was biased based on the available gene array chips at the time. And it really didn't look at the early time frame. They got one sample within the first 12 hours, but couldn't tell you what was happening over the first 12 to 24 hours very accurately. So we've taken a slightly different view in our work, and that is that we focus really on, when we started this work, we felt it was very important to focus on what was happening within the first few hours, 12 to 24 hours, and that by understanding what was happening here, we thought we could identify possible targets that would allow us to bring this response back into a safer range. And that's still a paradigm that is unproven in the trauma world. It's not known whether you can interrupt through early intervention, but it's still, I think, a reasonable goal. But this was our mindset for the study that I'm going to show you, so you know where we at least started from a conceptual point of view. And as Jörn mentioned, have taken a triphasic approach. We think it's always important to start with observations in humans because of the complexity of the human response, and because of the fact that many of the things we see in humans are just too difficult to model in experimental systems. But based on these observations, we would move towards mechanistic studies in experimental systems. And we've used mostly mouse genetic models to pursue this, and then apply systems biology analysis to try to identify pathways that may be important, or at least help us understand where we should be developing new hypotheses. And that's worked pretty well. So based on that, and based on the work of others, I'm going to go out on a limb here and show you what we currently use as a framework, a very simplified framework. It's going to look complex by the time I get done with it, but I assure you this is a very simplified framework for what we think might be happening in the immune response following moderate to severe injury in humans. And I'm going to start on the top with trauma, shock, and basically an immune response. And the endpoint is this clinical endpoint that I've been talking about, persistent critical illness and susceptibility to infection. So again, it's a composite, some of it's speculation, and I would view it as a framework. So clearly there are initiating events, and we believe these initiating events lead to a DAMP surge, a release of damage-associated molecular pattern molecules. There's clearly an associated, and we don't know exactly how it's interrelated, activation of coagulation and complement. That's established in both clinical and experimental settings. Patients who do poorly after injury all have some degree of shock, so there's going to be some degree of tissue stress and damage taking place, and this is all happening within seconds to minutes, so this is not something necessarily happening over hours. We think that this tissue stress and damage feeds back and results in a greater release of DAMPs. So DAMPs not only get released by the mechanical stress of the trauma, but get released by cell activation processes that happen that occur within minutes to hours. That's been established in experimental systems. So then the next step is cell activation. And DAMPs clearly can activate pattern recognition receptors. What is not often appreciated is that this takes place both on non-immune cells, such as epithelial cells, platelets, and endothelial cells should be up there as well, but also immune cells. And there are key sensors such as macrophages, dendritic cells, and neutrophils, and you could probably put your favorite immune cell up there as well. And I would suggest even lymphocytes express pattern recognition receptors, but just, again, to get a framework on the page, pattern recognition receptors activation should be viewed as a very early step in this immune response. Other cells clearly get engaged early. How they get engaged and what they do isn't known in the clinical setting, but experimental work would support that they're active, such as iNKT cells, B cells. We've shown, for example, that deletion of iNKT cells actually exaggerates the injury response. It looks like you need iNKT cells to suppress some of the tissue damage. Gamma delta T cells, NK cells, perhaps drive this forward, but how they're activated in the setting of trauma isn't known. So you've got some innate-like lymphoid cells clearly getting involved within minutes. And we began to think a lot about the role of epithelial cell stress, the production of epithelial-derived cytokines such as IL-25, 33, and TSLP, and the role of these recently described innate lymphoid cells, which sit within the tissues. You would never find out what's going on with these cells by sampling what's going on in the circulation. These are cells which are basically sitting in the tissue waiting to respond to epithelial stress and produce cytokines that I'll talk about. There's a limited amount of proof for their involvement. Mast cells are also activated by these same cytokines, and we do know that mast cells are activated early, so that's something we need to think about. But we also know that pattern recognition receptor activation leads to the production of chemokines, which can influence cell leukocyte trafficking, but also regulatory cytokines. And Urim's talked about some of these, such as IL-6, IL-23, being produced. So this needs to force us to think about some of the targets of these regulatory cytokines, such as Tregs, Th2 cells. Jim Leder, John Manick have done impressive work on these cell types, and we know that Tregs are activated, but in many of these models, they're looked at, several days after the injury. Whether they're activated within hours isn't known. Maybe Jim can shed some light on that, but it's thought to be driven by sets of regulatory cytokines, and this is probably all part of an adaptive response, and Jim's shown that nicely. If you take Tregs away, you actually get an exaggerated and dangerous inflammatory response. Th2 polarization takes place in just about every trauma model, and the consequences of that really isn't very well understood. But we have to anticipate that many of these pathways produce protective molecules, such as IL-22, we know is protective to the epithelium, that results in largely in resolution in the patients that do well. So what about the patients that don't do well? What might be happening? Well, we can engage other kinds of pathways. We know these regulatory cytokines will drive Th17 cells. We know IL-17 is up in patients that are severely injured. We don't know much about Th17 cell function within the trauma setting. But these pathways, plus pathways regulated by chemokines, can all result in excessive inflammation that can drive tissue damage and stress forward. We know that if you look at what we refer to as organ dysfunction, what we're really talking about is endothelial activation and barrier dysfunction combined with epithelial barrier dysfunction. That's what we see when we see organ dysfunction. So we get persistent organ injury, something that lasts more than a few hours, goes on to several days in patients that will go on to do poorly, and that we get immune dysregulation, which is really part of this excessive inflammation paradigm. And this could be driven by, again, even CD4 cell subsets with mediators produced, say, by Th2 or Th17 cells driving immune dysregulation. I put stronger arrows here for Th2 and Th17. Some of it's speculation, but the Th2 shift is clearly established in injury models. And ultimately, this combination of organ dysfunction, immune dysregulation, is what appears to render patients susceptible to this persistent critical illness and NI. Some of this is just a framework. Some of it's shown, but it's a starting place for the discussion. And I would emphasize that this is highly simplified. Even though it looks complicated, any one of these steps, you could put in dozens of additional interacting steps. And I'll show at least one example of that. So some of this viewpoint comes from our biomarker study that Yoram's introduced, which was really a massive effort. We accumulated nearly 500 blunt trauma patients. Admission criteria, they had to be adults. We used blunt just to be consistent, try to get some homogeneity. The ticket to get into the study was ICU admission plus consent. So, and we could get consent up to 24 hours later, so we could start the sampling right away, but had to discard, and we really had to discard almost no samples. We excluded isolated head injury because head injury will drive outcome more than anything else if it's severe. And patients needed to survive at least a day, and again we excluded penetrating trauma. And our goal was several in terms of using biomarkers. We viewed biomarkers as a discovery tool. We thought we could identify the dynamic relationship and mediator profiles. We could implicate novel components of the immune system by only being biased by the mediators that we were able to measure, and then identify potential therapeutic targets. We also thought that biomarkers would be useful for patient stratification, and this has been a major emphasis more recently. We thought we could potentially identify subsets early to modify therapy. I mean, if you had a biomarker on presentation that would tell you which way a patient's going to go, that would be very helpful. And then we could stratify patients early for interventional trials, and these are some of the directions we're going now with the work I'm showing you. And this is the paper that's already been mentioned that we published in Annals of Surgery and represented a considerable amount of effort on Rami Names's part, and it's been a full partnership with Yoram. We've shared ideas for over a decade, and many of our clinical colleagues are shown here as well. So this represented, again, analysis of 500 patients, at least 10 samples per patient, and up to 26 biomarkers, so huge amount of data. So many of you who are not in this field will, well, why is it ending up in such a journal like Annals of Surgery? Annals of Surgery is the highest impact factor journal in surgery. It's about 7. But I can promise you, and we sent this work to other journals, more prominent journals, without being able to demonstrate that we changed the outcome in the patients. They weren't interested in it. I mean, this is what happens to work in critical illness all the time. Just doing this study was a Herculean effort, but you're not going to get it published in a mainstream high-end journal unless you change outcome, which would be another decade's worth of work. Just a little editorial comment about the challenges we face in this field. So for the first part of the study, we measured at least 26 circulating cytokine, chemokine biomarkers. We focused on early time points, and this was key, at least 3 blood draws within the first 24 hours and then daily out to 7 days. And for this study, we correlated it with nosocomial infection, which I'll show you nicely is a nice surrogate for persistent critical illness. In other words, staying in the intensive care unit for a long time. This is what the infection, no infection, an infection was detected by standard CDC criteria. Shows the age of the 2 groups. We had about a 27% incidence of infection. You can see the male-female distribution. Those that get infection spend, again, about 2 weeks in the ICU compared to 4 days for those that don't, about 10 days on the ventilator. The problem here, however, is as you would predict, those that go on to get infection have a severe degree of injury, measured by injury severity score. So this is a standard scoring system. Doesn't detect all injuries, but it's calculated after discharge, because by then you know all the injuries. So ISS of 25, anything over 15, just to give you a reference point, it's considered a pretty significant injury. So 24, 25 is kind of moderate to severe. So you could just look at this and say, well, of course, if a patient's more severely injured, they're more likely to get an infection. Yoram, Rami, and I discussed this at length and decided to pursue this stringent one-to-one matching strategy to see if we could identify biological differences in patients with a similar degree of injury. So this is the total cohorts represented by this circle. Here's the 127 in which infection was detected. We found 44 patients that had no transfusions or no major operations. They all had some procedures done, but no major, because these are immune modifiers. So we wanted to pull these known immune modifiers out, at least for this fairly pure study. And we matched them with 44 patients that looked the same, except didn't get infection. And I'll show you the match. I mean, in terms of everything, they look the same. Same age, same number of men and women, same ISS, and then you can see we still get this segregation into this group, demonstrating PCI, persistent critical illness. The other group, they all go to the ICU. They're sick enough to go to the ICU. If you're going to the ICU after injury, you've got pretty significant injury. Again, 10 days on the ventilator. Many of these didn't even go on the ventilator, and much longer in the hospital. So this group is sick. This group injured to a similar degree, but not quite as sick. They both came in with similar degrees of elevations in lactate, which shows they both had some systemic hypoperfusion. A lactate above 2 shows that you're in at least some degree of shock. And this is the kinds of infections we saw, about half pneumonias, a third urinary tract, about 20% just detected as bloodborne infection. And this is a very important point. The infections are detected about a week after admission. So don't think of the infection as driving the processes that I'm going to talk about. This is something that represents a consequence of those processes, which is an immune deficit that allows microbes to escape into their body compartments and not be removed. So the infections are showing up at a week or beyond. They're probably there earlier, but the clinicians detect them at about a week. And this is a very important differentiation as well. So this is looking at Marshall score, which is one of the 3 or 4 major scoring systems that just look at organ dysfunction. Look at, whether there's elevation in the liver function enzymes as an example to estimate whether there's some liver dysfunction going on. They both come in with a slight elevation in their organ failure scores, but the group that doesn't get an infection just gets better. The other group actually worsens, and we now know that organ failure scores usually peak at around day 3, and you get this kind of football-shaped difference here. But when they get their infections, they're not in severe organ failure. That organ failure measure, these parameters of organ failure are peaking earlier on. So when you drill down into the injury patterns, and this is an important point, we do see a slightly higher but significant increase in chest injuries and face injuries in those that got infection. This has led us to believe that ISS, and this is something that would be debated among trauma surgeons, really isn't all that accurate as an estimate of injury severity. So we've started a collaboration with colleagues at Indiana University to come up with a better way to define the injury burden, and we think we have a better strategy for this than just to use what's been used for many years, which is this scoring system. These are the mediators that were measured on the first round based on a multiplex Luminex assay that Urim and Derek spent a long time validating, and we believe that it gives us pretty accurate results. And we found 4 distinct patterns that differentiated the infection from the no infection group. Again, we pored over this data for a long time, and much of this was done by Rami. We found there were mediators that were elevated upon admission and to a significant degree in both cohorts. Red is infection, blue dotted is no infection. And then they drifted downward. And I'll show you some specifics on this, but you can see these were largely chemokines plus IL-10. We found mediators that were markedly elevated even on the very first blood draw. So this is when the trauma patient shows up in the trauma room, you draw blood. That can be anywhere from 30 minutes to 4 hours after injury, depending on how long it took them to get there. And these mediators, for the most part, drifted down over the first 24 hours and included things you would expect like IL-6, but MCP1 and IL-1 receptor antagonist. And then this very interesting pattern was the accumulation in the serum or in the blood of many cytokines that would be associated with the lymphoid compartment. So IL-7, 5, 17, 4, 13. We'll go over these in more detail. And they rise in the second 12 to 24 hours, but only in the group that goes on to get infection. And then pattern D were some cytokines that typically more associated with sepsis, like IL-1, TNF-beta, elevated in the infection group, but never got very high and kind of drifted up towards the time that they began to experience infection. So here's some examples. Here's that pattern A, elevated in both. So there seems to be a common injury cassette, if you will. If you're injured to a certain degree, you're going to get chemokine elevations, at least certain chemokine elevations that go up. And it shows that both groups actually were pretty significantly injured, but no differentiation. Here's IL-6, even first blood draw, about 5 times higher in the group that would go on to have infection down here. MCP1 was even cleaner. I mean, it really segregates the groups very nicely, but drifts down within the first 12 to 24 hours. Here's this third group. Here's IL-17A. Again, not elevated on admission, but going up in the second 12 hours and then staying up. Pretty wide standard errors, but that's consistent with what you see in such a heterogeneous group of patients. And IL-7, which has turned out to be a really good differentiator across a number of our analyses. We'll talk about that a little bit more. And then these sepsis-associated cytokines that really never got very high. I mean, these are very low levels, but did show a difference between the groups. And I think this reflects an important limitation of this kind of study, but you know, we're only sampling the circulating compartment. We can't tell you what's happening in the tissues. And actually, if we did an area under the curve analysis for the first 24 hours, we found that 19 of the 26 were significantly elevated in the infection group, and that's shown here, and I'll come back to this list a few times. And then we've taken a number of systems analysis, and this is just what the dynamic network analysis that Urim talked about a little earlier looks like for this group over time, where we looked at the network connectivity using this time-based approach, and in this case, the correlation coefficient between the nodes was set at 0.7, so you can set it anywhere you want. But it provides a visual of what seems to be, might be happening. And so the top is the infection group, the bottom is the no infection group, and what should strike you immediately is that this is a far more complex interconnected system than the no infection group. This simplifies over time. Again, this is a little different than the survivor, non-survivor. That represented cohorts that were much more severely injured. And the other thing that happens is that the network becomes more complicated in the group that goes on to develop persistent critical illness. So now come back to this figure and how do we try to begin to dissect what's happening. I have no problem taking a more reductionist view of things because I think that's the only way you're going to sort out mechanism. So we began to look at what we thought might be the major drivers within at least this framework and paradigm, and what we were seeing within our cytokine chemokine signatures. So as I'll show you, and consistent with data, we thought that there might be a series of checkpoints or rheostats, in other words, things that if dialed up or down might be driving some of the biology that we're seeing. And we proposed that one of the major checkpoints would be the magnitude of the initial DAMP surge, HMGB1, mitochondrial DNA. There's probably dozens of DAMPs that play a role here, and that this would be a key driver, and I'll show you some data for that. That pattern recognition receptor sensitivity would be important. So how, based on an individual's genetics, how much might the differences in our pattern recognition receptor responses drive some of this response? We think that another sort of inflection point is the development of excessive or sustained barrier damage or dysfunction, driving a lot of not just the organ function but also immunology. And then as I'll show you, it looks like the cytokines suggest that there's an engagement of type 2 and type 3 immune responses. And we'll define what those are, but most of the immunologists in the field would know what these are, but you wouldn't necessarily think about type 2 and type 3 immunity as it relates to trauma. You'd be thinking about it as it relates to antimicrobial responses. And for each step, there's likely to be factors that determine how much this particular component plays a role. For example, injury severity and pattern for magnitude of the DAMP surge. genetic differences, gender-based differences for pattern receptor sensitivity, pre-existing disease, and perhaps immune memory for something like type 2 and type 3 immune responses. You can make your own list. But in terms of some examples that the magnitude of the damp surge is important, this is looking at our study, looking at HMGB1, and you can see HMGB1 levels are twice as high on admission in the patients that go on to get infection compared to those that don't. And consistent with our own previous work and the work of others, the HMGB1 is kind of the prototypical damp, is elevated early and then drops off fairly quickly. So it is consistent with that pattern. In terms of pattern receptor sensitivity, we've looked at a polymorphism in IRAK1, which is a common signaling intermediate to essentially all of the toll-like receptors at least. So just about everything goes through toll-like receptors. And this particular gain of function polymorphism has been shown to be associated with worse outcomes in sepsis. In Caucasians, it's expressed in about 20% of people, and it's X-linked. So if you're a male, you're much more likely to manifest the polymorphism. And of course, males, by most studies, do worse than females. And Jason Sperry and I published this paper last year showing that in a relatively small number of patients that carry the variant, 32 out of 275 showed a dramatic increase in MOF and mortality. So this is kind of some data saying that your genetics in terms of your pattern receptor sensitivity may drive some of the extent of the response, and we're correlating this with some of the cytokine signatures now. So now come back to the this list of 19 mediators. What should strike you is that 5 out of the top 6 mediators showing the greatest fold difference in this area under the curve analysis were cytokines associated with either lymphocyte regulation or produced predominantly by lymphocytes. And so I take you to the paradigm for at least CD4 T cell polarization, and we begin to look at where the mediators line up. For example, IL-4 drives a Th2 response, and then Th2 cells, for the most part, at least their cytokine signatures, IL-4, 5, and 13. Looking at Th17 cells, driven by cytokines that we've all looked at for years, but not necessarily thought of in terms of the context of how they might play a role in CD4 polarization in trauma and sepsis, because we just haven't thought about that very much. As you can see, IL-6 and 23, TGF-beta, these are the 3 cytokines that are critical for the differentiation and expansion and stabilization of Th17 cells, which then produce IL-17, IL-21, and IL-22. So we're going to look at those signatures in these patients. And here is what we see for IL-5, 4, and 13, and I'll show you the graphs as well. But in terms of our mediators showing the greatest fold difference, 5, 4, and 13 are all in the top 10. So it looks like a type 2 signature in the patients that develop persistent critical illness. And products of Th17 cells, IL-17A, GM-CSF also lined up here as well. And I'll show you some data for IL-22 that we went back and looked at IL-22. But what caught our eye was the appearance of IL-7 and IL-15. And you look at what these 2 cytokines do, they do a lot of things. But among the things these cytokines do is drive differentiation of cells towards innate lymphoid cells. So innate lymphoid cells come from a common precursor, but based on a number of factors that aren't entirely understood, can drive these cells, which reside in the tissue. So there's 3 groups, group 1, group 2, group 3. Group 1 ILCs, Th1-like for the most part. NK cells fall under this group. So you have regulatory ILCs, such as 1, 2, and 3, and then NK cells. And then the type 2 ILCs respond to epithelial cytokines that I've already mentioned, 25, 33, TSLP, to produce cytokines, the exact same cytokines that Th2 cells produce. But these cells are sitting in the tissue ready to be activated very quickly. ILC3s look a lot like Th17 cells. They produce IL-17, 22 interferon in response to perhaps epithelial cytokines, but as I'll speculate, also IL-23. So this is the paradigm now for ILC activation. And these pathways have been worked out predominantly in the setting of more chronic processes such as parasite infection, allergic responses, and some viral infections, where the epithelium is the driver through the production of these cytokines that I've been talking about. And this is the paradigm for ILC2s, which can then produce cytokines such as IL-13, which will drive dendritic cell activation, conversion to M2 type of phenotypes. And these cells can also drive Th2 differentiation. So you can have a link between ILCs and CD4 polarization through epithelial stress, epithelial damage, which of course is what we see in our trauma patients. So we've put together kind of a sub-paradigm for this pathways to these type 3 and type 2 immune responses, where again, driven by damp surge or perhaps non-PRR activation, we get activation of immune cells for the production of regulatory cytokines, and things like IL-6 can come from other sources. But we know that these cytokines are elevated in our patients, and they can drive Th17 production. They can also drive IL-3 activation. But epithelial cytokines can drive ILC3s, maybe, but more importantly can drive type 2 immune responses, as well as Th2 cells. And these are defined based on the cytokine signature. So we're proposing that maybe we should be talking about this immunology in a little more mainstream context, and not just in terms of a specific cell type, but the kind of immunology that you're getting. So we know that mast cells, for example, can produce IL-4 and 5, MDSCs as well, not just lymphocytes. And this is a diagram that Rami put together just in the last week as we've expanded our analysis to additional cytokines. So thinking about it in the context of regulatory cytokines and then types of immune cytokine signatures, we've lined it up in that way. So we've added to our IL-6 measurement that I've shown you here earlier for the infection versus no infection, IL-23, a major driver of both ILC3s as well as Th17s. And you can see IL-23 is markedly elevated and elevated early in the patients that go on to develop a complicated course. IL-4 kind of fits in that third pattern where it's not up so early, and therefore how much it's playing an early role as an effector versus, or as a regulator versus an effector isn't clear. We went back and looked at epithelial regulatory cytokines, 25 and 33, both up. 33 is up on admission. So we've got a lot of epithelial stress going on. And of course this will drive ILC activation. And then the type 2 cytokines we've already mentioned. IL-5 climbs up a little later, IL-13 climbs up a little later, but clearly up within the first 12 to 24 hours. It's not taking days to detect a type 2 immune response signature. And then the type 3 cytokines, IL-17 I already showed you. We've now added IL-22, which interestingly is up even earlier. But we know IL-22 can come from a number of different sources other than just Th17 or Th22 cells. So it looks like we have the cytokine signature consistent with that paradigm. So we can perhaps move in that direction. And we've gone back now. This is where you do take the iterative strategy and begun to look for ILC2 activation in our trauma model. This is work done by Shuhua Chen with Rosie Hoffman. And we look at lymphocyte lineage negative populations from the lung. And you can see, looking at this population here, which has the markers for ILC2s, a near doubling over the first 24 hours in our hemorrhagic shock trauma model in mice. This is very new preliminary data that we're excited about. This takes a lot of work to phenotype these cells, but I think it's the amount of work that's necessary to provide support for the concept. So I'll stop here. Here are the conclusions. Trauma victims experience similar injury patterns, and experiencing similar injury patterns can exhibit markedly different clinical trajectories. And that's been known for decades, and we're just finding better ways to describe that. Divergent clinical trajectories are manifested not only by the magnitude and duration, so that's the data from the last decade. Magnitude and duration is important, but it's also important to think about the components of the immune system that are engaged, and they do appear to be different between the two patient populations. Persistent critical illness is associated with engagement of lymphoid components, at least measured in these studies by what appear to be type 3 and this should be type 2 and type 3 cytokine responses. Persistent critical illnesses are associated with evidence of persistent, early and persistent epithelial stress and cytokine production. If you were to do a Medline search for IL-25, 33, or TSLP with trauma or injury, you come up with no papers. There's nothing published in this domain. The magnitude of the initial DAMP surge may be an important contributing factor. We believe that. And genetic determinants of the host response are likely to contribute to the magnitude and nature of the trauma-induced DAMP response or injury response. So I think I'll stop there, and thank you for your attention. And again, thank you for this opportunity to show what we've been doing.